Hello everyone, welcome back on this platform. Today I'm here with this fourth part of this questionnaire of MSc Botany Entrance Exam 2022. In this part I will discuss questions from ecology, plant physiology and plant anatomy. So let's dive in. So the first question for today is continuous increase in concentration of toxic chemical in food chain is called. Our options are biomagnification, bioleaching, biodegradation, biodeterioration. So its right answer is biomagnification. Bio means living, magnification means increase in concentration of certain pollutant or chemical. So when the food passes from one trophic level to the next trophic level, its concentration gets increased. So that increase in concentration of these chemicals, pesticides or pollutants in each trophic level is known as biomagnification. So from mayfly to human, its concentration is increasing from passing from each trophic level. So this increase in concentration is known as biomagnification. For example, we have certain chemicals or pesticides that gets inside our body through inhalation, ingestion. So that can cause serious health concern, like some chemicals have cytotoxic effect or a genotoxic effect that can cause carcinogenic diseases in the humans. Then they also affect our immune system, respiratory tract, nervous system, reproductive system, or bloodstream. So these chemicals are very toxic and its concentration gets increased in each trophic level when they pass in the food chain, and this increase in concentration is known as biomagnification. The next comes bioleaching. So this is a technique by which we obtain certain precious metals from the ores by using certain living organism, like we have bacteria or archaebacteria. So that is known as bioleaching. So bioleaching or biomining is a process in mining and hydrometallurgy, that is natural process of interaction between microbes and minerals that extract valuable metals from our low-grade ore with the help of microorganisms such as bacteria and archaea. So that process is known as bioleaching. Next comes biodegradation. So it involves breakdown of certain organic substances with the help of certain microbes is known as biodegradation. So it involves biodeterioration, then biofragmentation, assimilation, and mineralization. Next comes biodeterioration. So biodeterioration or biodegradation is almost similar terms. So in which material can be defined as any undesirable change in the properties of material caused by vital activities of biological agencies of organisms. So that breakdown, or we can say change in chemical properties of certain organic matter with the help of microbes, is known as biodeterioration. The next question is, which of the following is released by automobile in the air? Our options are BHC, PCB, peroxyacyl nitrate, or fluorides. So this is a question from pollution. So its right answer is this peroxyacyl nitrate, that is abbreviated as PAN. So this is a secondary pollutant which is formed after the release of oxides of nitrogen from the vehicles that gets combined with oxygen to produce this a secondary dangerous pollutant that is called peroxyacyl nitrate. So here we can see from this image there are many automobiles or any other industries from which this volatile organic compounds gets into the air, which are like we have oxide of nitrogen which gets into the air, and these are considered as primary pollutant which directly comes from source, then they combines with oxygen and they will produce secondary pollutants, like we have ozone in troposphere is a dangerous pollutant. Then we have PANs, that is peroxyacyl nitrate. So primary air pollutants, we have different categories. Basically we have two categories of pollutants, that is primary air pollutant and secondary air pollutant. So these are the poisonous gases and undesirable substances that are released by burning of fossil fuels, and these substances are called as primary air pollutant, which includes the soot particles which are released from unburned fuel. Then we have sulfur dioxide, benzopyrene released from the cigarette smoke. Then we have ammonia oxide of nitrogen and carbon monoxide. So these all are then lead. These all are primary pollutants. The next category of pollutant is secondary air pollutants. So secondary air pollutants are poisonous substances formed from primary air pollutants, or even their primary air pollutants. Say in bright sunlight, nitrogen oxide, hydrocarbons, and oxygen interacts to produce more powerful photochemical oxidants, like we have this one, that is ozone, which is a very dangerous pollutant in troposphere. Then we have this peroxyacyl nitrate that is formed from the oxides of nitrogen. Then we have aldehyde, sulfuric acid peroxides, etc. So all these constitute photochemical smoke, which retard photosynthesis in plants. So we have two categories of air pollutants, that is primary air pollutant and secondary air pollutant. And the pollutant that is released from automobile is peroxyacyl nitrate, which is a secondary pollutant which is formed after the combination of oxides of nitrogen with oxygen. The next question is from anatomy, that is periderm is composed of, our options are cork, cambium and bark, then cork, cambium and secondary cortex, cork, cambium and secondary cortex and cork, then cork, bark and secondary cortex. So its right answer is cork, cambium, secondary cortex and cork. So these three layers leads to formation of this layer that is known as periderm. It is formed during the secondary growth in plants. So in the cortex region, some of the parenchymatous cells becomes meristematic and they will form the cambium, which is known as cork cambium, which is also known as phallogen. Then this phallogen produces some thick-walled cells towards the outer side, which are dead at maturity and have narrow lumen. So these cells are known as cork cells. They provide protection, and cork is also known as phallum. इसे phallum के नाम से भी जानते हैं. Then this cork cambium towards the inner side, it produces the living thin-walled cells, which are parenchymatous cells, to form structure which is known as secondary cortex, and it is also known as phalloderm. इसको हम इस term से भी जानते हैं, that is phalloderm. And these three layers combine to form this structure which is known as periderm. So it consists of this cork cambium, then it contains this outer layer which is called as cork cells, then inner there is secondary cortex. So these three layers produces these three layers form this combined layer that is called periderm. And bark is a non-technical term that includes tissue from phloem onwards. So this is found during secondary growth. So cork, then it contains this bark. Bark include the tissue, including primary phloem. The next question is, in a national park, protection provided to, our options are entire ecosystem, flora and fauna, fauna only, and flora only. So its right answer is entire ecosystem. So national parks are the protected areas which are involved in protection of entire ecosystem. Means it include all the flora and fauna along with some historical buildings or any other statues. Here human activities are restricted, so no human activities like grazing, timber collection is allowed here. So we have different protected areas, like we have wildlife sanctuary, biosphere reserve and national park. So they have some differences. So wildlife sanctuary are protected area which are specifically meant for the conservation of certain animals. Here there are less restrictions, so human activities are allowed to some extent, like we can take our domestic animals for grazing there, then we can collect timber. So it includes category 4 types of protected areas. So it is specifically meant for preservation of animals, insects or birds. While we have the next is biosphere reserves. These here the condition restrictions are moderate. So biosphere reserve contain different layers, zones. So it contains the zones here human activities are allowed. Then we have buffer zone where some sort of human activities are allowed, and core zone where no human activities are allowed. So this comes under this program that is called Man and Biosphere program, launched by UNESCO. Then it is used for protection of the biodiversity of our designated area. Next we have national parks. So there are heavy restrictions, human activities are not allowed. It falls under the category 2 of protected area. Here flora, fauna, landscape, historical objects, all are protected. So entire ecosystem is protected here. So this is the list of some important national parks, which include the Hemis National Park, which is the largest national park of India, which is located in Jammu and Kashmir. The next we have Guru Ghasidas Sanjay National Park, that is present in Chhattisgarh. The next we have Desert National Park, that is present in Rajasthan. Then Gir Forest National Park, located in Gujarat. Then we have Gangotri National Park, which is present in Uttarakhand. In addition to this, we have the Jim Corbett National Park, which was the first National Park in India. So that is also located in Uttarakhand. Then we have Sundarban National Park that is present in West Bengal. Then we have Nam Dafa National Park that is present in Arunachal Pradesh. Jim Corbett National Park again in Uttarakhand. Then we have this Kanchenjunga National Park that is present in Sikkim. And next question is Koina Sanctuary is located in. Our options are Maharashtra, Karnataka, Assam, or Andhra Pradesh. So its right answer is Maharashtra. So this wildlife sanctuary is located in Maharashtra, that is Koina Wildlife Sanctuary. In addition to this, we have different wildlife sanctuaries in Maharashtra, which include Nagzira Wildlife Sanctuary. Then we have the Doba Andhri Tiger Project. Then we have Karnala Bird Sanctuary here. Then next is Bhim Shankar Wildlife Sanctuary, Myani Bird Sanctuary, Sagareshwar Wildlife Sanctuary, and Radha Magri Wildlife Sanctuary. So these are different wildlife sanctuaries which are present in Maharashtra. Next question is from plant anatomy. Secondary growth is Anamylis and our options are Dracaena, Anoa, Helianthus, or Pinus. So its right answer is Dracaena, which is a monocot which shows secondary growth. As we know that monocot lacks cambium, so presence of cambium in monocot is itself anomalous secondary growth or anomalous growth. So this is the plant Dracaena. So this shows the presence of this cambium layer, which produces secondary parenchyma towards the outer side, and inner side produces vascular bundles, which include the amphivessel vascular bundles. It means it contains vessels or phloem towards inner side and surrounded by xylem. So these are concentric vascular bundles. In addition to this, it produces the tissue which is known as conjunctive tissue. So presence of these vascular bundles along with this cambium is known as anomalous secondary growth in case of Dracaena. So due to lack of cambium in monocotyledons, secondary growth is absent, but exceptionally secondary growth takes place in some monocotyledons, like we have palms, yucca, and Dracaena. So Dracaena shows this anomalous secondary thickening. The next question is who proposed Z scheme of light reaction? Z scheme is a term used for non-cyclic photophosphorylation. So it is the process of converting light energy into chemical form of energy in the form of assimilatory power, that is ATP or NADPH. So the name of scientists are Kelvin and Benson, Arnon, and we have Emerson, Hill and Bendel. So its right answer is Hill and Bendel. These were the two scientists who proposed this scheme that is known as Z scheme. So here in this non-cyclic photophosphorylation, the electrons move from photosystem 2 to photosystem 1, and they will not come back to the photosystem 2. So that's why this is non-cyclic photophosphorylation, means they will not return to its source. So that's why this is known as non-cyclic photophosphorylation. It involves formation of ATP from ADP in the presence of light. So that is called photophosphorylation. This is non-cyclic. So it involves photosystem 2 and photosystem 1. Photosystem 2 has one important complex which is known as OEC. OEC stands for oxygen evolving complex, which is made up of manganese and chloride ion. So they help in splitting of water and release of electron. So this acts as a strong oxidant. So it will absorb light, photons of light, and gets into excited state. Then electron are transferred to the pheophytin. Then we have plastoquinone through a series of these mobile electron carriers, which include cytochrome B6 and F. During this step of transfer of electron, ATP is produced. Then we have this copper containing mobile electron carrier, that is blastocyanine. It will send the electron to photosystem 1. इसके बाद जो electrons है photosystem 1 पे आते हैं. Again this photosystem 1 is strong reducing agent or strong reductant, and it absorbs at a wavelength of 700 nanometer. It will absorb the photons or light and gets into excited state. Then it is associated with this ferredoxin reduction center, which will carry the electrons to the final acceptor of electron, that is NADP, and it is converted into NADPH. It will involve reduction of this NADP to produce this assimilatory power, that is ATP and NADPH. So electrons start moving from photosystem 2 and ends up here. So they will not return back to the source. That's why this is known as non-cyclic photophosphorylation. And this was explained by Hill and Benton. So Z scheme of photosynthesis was proposed by Hill and Benton. Next question is again from physiology. So the red drop phenomenon is due to disruption of the photochemical activity of our options are photosystem 1, 2, photosystem 1 and 2, and carotenoids. So its right answer is photosystem 2. So Emerson observed that when a light of red region is provided to the chlorophyll, so there was a sharp decrease in rate of photosynthesis. So that sharp decrease of the red region of electromagnetic spectrum is known as red dwarf effect. So that decrease in photosynthesis is known as red dwarf effect. So that leads to discovery of other photosystem, that is photosystem 1, when light of two different wavelength, that is red and far red light, is provided simultaneously. So he observed there was an enhancement in the rate of photosynthesis, which leads to discovery of another photosystem, that is photosystem 1, which lies in the far red region of the electromagnetic spectrum. So this disruption of photosynthesis due to the disruption of photosystem 2. So this decrease in rate of photosynthesis when the light of red region or above red region is provided to the chlorophyll. So this sharp decrease in the rate of photosynthesis is known as red dwarf effect, and this was observed by Emerson, and it is named as Emerson red dwarf effect. When the light of red and far red light is provided simultaneously, so there was an enhancement in the rate of photosynthesis. So that enhancement is known as Emerson's enhancement effect. That leads to discovery of another photosystem, that is photosystem 1. So from here we know that plants have two photosystems, that is photosystem 2 and photosystem 1. Photosystem 2 absorb the light of red region of electromagnetic spectrum, and photosystem 1 absorb the light of far red region of electromagnetic spectrum. The next question is, little leaf disease is caused by the deficiency of which mineral element? We have zinc, copper, manganese, or molybdenum. So its right answer is zinc. So zinc deficiency causes little leaf disease in plants. Like we have one disease that is called little leaf of brinjal. It is also caused by MLO, that is mycoplasma like organism. So in addition to this, zinc deficiency also causes this disease, which is known as little leaf disease. Copper deficiency causes one disease that is known as exanthema. In this disease, plants secrete some exudate like glue. Then manganese deficiency. Manganese is an important component of this OEC, that is oxygen evolving complex. So plant is unable to do the process of photosynthesis. Then molybdenum deficiency, it is again the important component of nitrogen fixing bacteria. So it altered the nitrogen fixation process. This is the table which shows different diseases or symptoms caused by deficiency of certain mineral elements. First of all, chlorosis, that is the yellowing of leaves, that is known as chlorosis. So the main pigments which are involved in this, nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum. Then intervenal chlorosis, it is caused by the deficiency of magnesium, iron, manganese, zinc. Marginal chlorosis, it is caused by deficiency of potassium. The necrosis is the death of tissue which leads to formation of black spots. So this occurs due to deficiency of magnesium, potassium, calcium, zinc, molybdenum, copper. Stunted growth, it is due to deficiency of the main minerals, which is NPK. In addition to this, it is also shown by the deficiency of calcium and sulfur. Then we have anthocyanin formation, that leads to formation of reddish coloration in the plant parts, that due to deficiency of nitrogen, phosphorus, magnesium, and sulfur. Then we have dieback of shoot, so or reclamation disease. These are two important diseases. Then we have this exanthema in citrus. So this is due to the deficiency of this mineral element, that is copper. Then hooked leaf tip, that is caused by deficiency of calcium. Little leaf disease is caused by the deficiency of zinc. Then brown heart of turnip and internal cork of apple is due to deficiency of boron. Boron plays also an important role in pollen grain germination. Next, whiptail disease of cauliflower, it is due to deficiency of molybdenum. Curl leaf margins is done due to deficiency. It occurs due to deficiency of potassium. These are certain some elements which shows these. Next question, which element is essential for root hair formation? Our options are zinc, copper, calcium, or sodium. So its right answer is calcium. So calcium plays very important role in the formation of root hairs. So this was all about for our today discussion. I hope you will get some idea from this presentation. Thank you. Have a nice day.